My name is Julia Desiato. I'm a senior pathobiology major at the University of Connecticut. Today, I will be presenting on the evolution and spread of rabies virus in Connecticut and its neighboring states. This project was conducted at the University of Connecticut in the Genomics and Molecular Epidemiology Research Lab in conjunction with the Connecticut Veterinary Medical Diagnostic Laboratory, known as the CVMDL. First of all, I would like to briefly talk about the background of rabies virus. Rabies virus is an enveloped RNA virus that belongs to the genus Lysovirus. It has a 12 kilobyte long non-segmented RNA genome encoding 5 viral proteins, N, P, M, G, and L. The disease is typically transmitted through the bite of an infected animal via saliva. Infections attack the central nervous system and may result in symptoms such as respiratory arrest, paralysis, encephalitis, coma, or fatality if the patient is not treated with post-exposure prophylaxis promptly after being exposed. Rabies virus has been circulating in the United States for decades after being identified in Florida in the 1940s. Prior to 1960, the majority of cases were seen in domestic animals. However, the introduction of pet vaccinations led to a decline in cases in domestic animals. The decrease in cases in domestic animals led to a decrease in human fatalities, and there were 127 cases of rabies in humans between the years 1960 to 2018 in the United States. Raccoon variant rabies virus migrated north from Florida into West Virginia in 1977 as raccoons were relocated to West Virginia for hunting. Since then, raccoon variant rabies virus has spread throughout the mid-Atlantic and up through the northeastern states and has been able to maintain a stable spread in wildlife. As you can see in the map here, the main species that host the virus in the United States are the skunk, the fox, the raccoon, and the mongoose. Rabies virus first appeared in Connecticut in 1991 during an outbreak in Fairfield County. Eventually, cases spread to the remaining 7 counties in the state. There have been a total of 3,421 cases throughout the state, with the highest numbers in Fairfield, New Haven, and Hartford counties. Data from the Connecticut Department of Public Health website on rabies virus cases going back to the year 2000 were collected and organized to assess which species have been maintaining the virus in Connecticut. Of those 3,421 positive cases, the majority of them are in raccoons and skunks. There is a species breakdown of 1,900 reported cases in raccoons, 822 cases in skunks, and the rest among 15 other species. Other wildlife, such as woodchucks, deer, and bobcat, also have been infected and make up the 3rd largest group of cases. Livestock and other companion animals have really low case numbers. Rabies cases in bats have been excluded from our analysis, since they are infected with a bat variant rabies virus rather than the raccoon variant samples that we have sequenced. Only limited genetic information on rabies viruses in Connecticut exists, and the population dynamics of circulating rabies viruses, potential drivers of virus changes in the states, remain uncertain. Rabies virus is typically transmitted through species-species interaction. However, cross-species emergence of the virus poses another public health concern. Enhanced surveillance and characterization of viruses circulating in Connecticut will highlight the evolution of the virus and cases of cross-species transmission in the New England area. Although rabies virus has not established a permanent transmission cycle to a new host species, cases throughout Connecticut must be tracked to predict potential host shifts and determine evolutionary changes in the genome that would contribute to its ability to persist in a new host. While rabies virus is endemic in Connecticut, there is not a strong understanding of the disease dynamics in the area, specifically the evolutionary history of the strains found in diseased animals. In this study, we conducted the genetic characterization of rabies viruses detected in Connecticut by full-length genome sequencing and comparative phylogenetic analysis. Samples for suspected rabies cases between the years 2017 and 2020 submitted to the CVMDL lab were used. Since rabies virus infects the central nervous system, a cross-section of brainstem tissue is typically assessed post-mortem to identify if rabies virus proteins are present. The presence of rabies virus in brain tissues was determined by using the direct fluorescent antibody test, also known as the DFA test, and confirmed by real-time RT-PCR. 63 total rabies-positive samples identified in the period of 2017 through March 2020 were sequenced. So far in 2020, only 2 samples were submitted for sequencing. After rabies virus was detected, multiplex RT-PCR was conducted to amplify the entire viral genome sequences. Whole genome sequencing was performed using the iSeq 100 Illumina platform. Before we analyzed the data, we checked the quality of the raw sequencing data using FastQC software. Trimmomatic, Bowtie2, and iVar software were used on the Galaxy server to trim, filter, and map the sequencing reads to a known reference rabies genome sequence. Mapped alignment software was used to align the newly obtained consensus sequences so that they could be run through phylogenetic analysis. We retrieved 916 North American rabies virus sequences on GenBake and then used an Elam dupes tool to prune down sequences with a 99.5% sequence identity level to filter out representative sequences and ended up with a total of 203 sequences. A maximum likelihood ML tree, and a Bayesian timescale tree was created using the RAXML and the BEAST package, respectively. This image here is the timescaled phylogenetic tree created with the 203 reference sequences from North America. The phylogenetic tree revealed 2 distinct clades with well-supported 7 different subclades of rabies virus in Connecticut. The colored portions represent the most well-supported subclades that were created based on the distribution of the Connecticut sequences. The long lengths of these branches from node to sample represent a gap in sequencing data. Clade 1 is divided into 3 subclades, 1A, 1B, and 1C. The 21 sequences in this clade are genetically similar to a New York sample. We estimated the time to most recent common ancestor, known as TMRCA, for these subclades. The TMRCA provides an estimate of when subclades emerged that allows the possible date of introduction to Connecticut of each subclade to be inferred. As shown in this table, subclade 1A has a node age of November 2010, 1B has a node age of November 2009, and 1C has a node age of June 2013. This clustering and the positioning of the samples in southwestern Connecticut reveals that the most likely genetic origin to these samples is the state of New York. In subclade 1A, there are 16 raccoon sequences. In subclade 1B, there are 3 samples, a bobcat, a raccoon, and a skunk. In subclade 1C, there are 2 raccoon samples. All samples lie west of the Connecticut River, which most likely suggests that the river acts as a barrier to the spread of clade 1 rabies virus. Clade 2 has 4 distinct subclades, 2A through 2D. These samples are spread throughout Connecticut in a variety of species, but mainly raccoons. Clades 2A and 2B clustered together with these 7 Vermont sequences from 2007 to 2009. We will look at the geographic distribution of each of the 4 subclades to identify any patterns in the groupings. As the TMRCA table shows, there is a broad time period in which these sequences diverged. Subclade 2D has a node age of January 1997, which is the earliest, and subclade 2A has a node age of July 2006, which is the latest. Clade 2A has a total of 21 samples. They are spread throughout the state of Connecticut and have clustered closely with the samples from Vermont. The subclade itself contained 1 sequence from Vermont in 2017. Clade 2B has a total of 15 samples spread between raccoons, skunks, felines, woodchucks, and a bobcat. For the most part, the samples are located on the eastern side of Connecticut and clustered with a sample in Rhode Island, as well as many Vermont samples. There is one outlier, which is the single sample to the west of the Connecticut River. Upon research into bobcat migratory patterns, it is possible that it was able to cross the Connecticut River, which could allow it to be so geographically distant from the other 14 samples. Another possibility is that since the samples clustered with samples from Vermont, their travels from north to south would not be impeded by the Connecticut River, which could have allowed the samples to separate. Both clades 2C and 2D have 4 samples each. For the most part, these samples are located along the Connecticut River and near the midline of Connecticut. Subclade 2C clustered near those same 7 Vermont samples as 2A and 2B. However, subclade 2D was a bit more genetically distant from the rest. Samples in this group clustered mainly with samples from Maine, but also some samples from Vermont, Massachusetts, New Hampshire, and New Brunswick, Canada. Overall, there were 3 different newly sequenced samples that did not cluster with any of the clades, which indicates that they may have been sporadically introduced into Connecticut. The 3 samples were from cow, raccoon, and white-tailed deer species. Geographically, they are also fairly distant. The estimated node ages, or most recent common ancestor, for each of the clades varies within a range from January 15, 1997, up through June 10, 2013, suggesting multiple lineages of viruses were introduced or emerged in different time points over the years, which is what has allowed their independent diversification into 7 distinct subclades circulating in Connecticut. Rabies virus data was organized on Tableau software to illustrate the geographic spread of the 70 sequences from Connecticut. This map illustrates the species of the sample in color based on their common name, and each point has a letter and number to indicate which subclade they belong to. As you can see, the green color representing raccoons has the highest number of samples throughout the state, followed by skunks. There is a concentration of cases in raccoons in the southwest corner of the state, which most likely suggests that there is an incursion of subclade 1 virus from New York. Based on the distribution of cases overlaid on a topographic map of the state, data most likely suggests that the Connecticut River and major roads such as Interstate 91 have prevented the spread of one lineage of the virus across the state. Phylogenetic evidence and geographical distribution show 7 different subdivisions of the virus throughout the state of Connecticut. Further sequencing and phylogeography analysis will be required to do better analysis of the disease dynamics of rabies virus in the state. We reconstructed this tree using only sequences from Connecticut to investigate the evolution of rabies in the state. The substitution rate was 2.17 times 10 to the negative 4th substitutions per site per year, which is similar to the substitution rate of rabies viruses in other regions. On the right is an image of the CRWAD plot. A GMRF Bayesian CRWAD plot illustrates the effective population size over time and represents the relative genetic diversity of rabies virus in Connecticut. The dark blue line represents the median estimate of genetic diversity over time, and the shaded regions are showing the 95% credible interval for genetic diversity. As you can see, genetic diversity has been steadily increasing until 2016, but the increased diversity is maintained after 2016. As illustrated by the 7 subclades, there are multiple different raccoon variant rabies viruses currently circulating in the state. The CRWAD plot shows that there is high genetic diversity of rabies virus in Connecticut that continues to diverge further. Currently, there are many different host species of the virus. However, raccoons are the main reservoir species in this area of the country and may be contributing to the maintenance of the 7 subclades within the state. Phylogenetic and geospatial analysis most likely suggests that the Connecticut River acts as a barrier to stop the spread of clade 1 rabies viruses, as all of the samples in the clade remain to the west of the Connecticut River. The relatively large time lapse between certain lineages of raccoon variant rabies viruses from other states shows how critical it is to perform whole genome sequencing in Connecticut. The gap in sequence collection between the Connecticut sequences and their closest common ancestor, as illustrated by the long branches in the phylogeny, shows that the cases were circulating in the state at this time, but have yet to be sequenced and are critical to establishing an evolutionary timeline of the virus. Currently, we are unable to differentiate how many incursions of raccoon variant rabies virus there have been from other states since it first entered Connecticut. For example, the clustering of clade 2 with 7 different raccoon samples from Vermont in between the years 2007 to 2008 suggests that this specific raccoon variant rabies virus could have been maintained somewhere in New England for close to a decade, but without sequence data, it is not possible to tell for certain. Through further sequencing, we hope to identify how rabies virus has remained stable throughout the state and to determine if there will be a newly emerging reservoir species for the virus. I would like to thank my advisor, Dr. Lee, and lab members, David and Scott, and also Dr. Rosati and staff at CVMDL. The study is funded by the UConn Office of the Vice President of Research's Research Excellence Program. Thank you.